thank you very much for attending my presentation on machine learning in biomedicine, from basic research to clinical translation. My name is Lars Kaderali. I'm professor for bioinformatics at Universitätsmedizin Greifswald and head of the Institute of Bioinformatics. Before I start with the presentation, I would actually start with a few words about myself, because I assume most of you do not know me. I studied computer science at the University of Cologne and did a PhD on high-dimensional regression in medicine, namely predicting whether a patient with neuroblastoma, a particular cancer, survives or not, based on gene expression profiles. Subsequently, I worked at Los Alamos National Laboratory in the United States. I did work at the German Cancer Research Center in Heidelberg at the Department of Bioinformatics, then headed a junior research group at the University of Heidelberg. From there, moved to a W2 professorship at the University Hospital in Dresden. And now, since 5 years roundabout, I am a professor of bioinformatics at the medical faculty of University of Greifswald. And in between, I had a few short stints in industry. I did work for about half a year as a software developer at Cell Corporation in Canada. I worked for a summer as an intern at ITK, that's a ISDN card development company in Germany, where I was again working as a software developer. I am still a partner in ISL GmbH, that's a company that focuses on IT security, and I'm still working as a consultant for Bayersdorf AG. I've been working on artificial intelligence in biomedicine since around about the year 2000, and over the years have been moving more and more into applications. The institute that I'm heading at University of Greifswald was founded with my move to Greifswald in 2015, 2016. It's part of the University Hospital and the medical faculty. We are 2 professors, approximately 20 staff, including postdocs, PhD students, bachelor, master students, and administrative staff. And most of them are funded through third-party grants, and most have a mathematics or computer science background. The institute is the former Institute of Biometry and Medical Informatics, so we are doing most of the statistical consulting and the clinical trial planning for members of the medical faculty, and we are also involved in teaching medical students in biometry and biostatistics, and also in bioinformatics, and that's to students of human medicine, human biology, and also biomathematics. On the research side, what do we do? On the one hand, we are developing and applying statistical and bioinformatics methods, mostly to large-scale omics data, that's high-throughput screening data, gene expression arrays, sequencing data, and of course, being at a medical faculty, also clinical data. Then part of the group is working on developing mathematical models of molecular and cellular processes. So, for example, we have developed models of how viruses spread in a population of cells, how they replicate inside cells, how they activate the immune response. So what we do there basically is quantitative dynamic understanding of cells, developing computational models, and then using these models to simulate processes occurring in a cell. And on the other hand, and this will be the topic of the presentation today, we apply methods from artificial intelligence to medical questions. And here we both develop and apply machine learning methods, in essence, to learn predictive models from data, integrating different types of data. So here really this is data-driven model learning. And I will have several examples for you of this today. And ultimately, we try to integrate these 3 different approaches. So basic statistics, bioinformatics, omics, mathematical modeling, and artificial intelligence to really understand health and disease using these statistical and computational approaches. And in the end, being at a medical faculty, our aim is to translate our research finding into clinically relevant approaches. Okay, and with that, I would like to come to the topic of my presentation, artificial intelligence in medicine. And this is a very much hyped topic today. There's also a lot of hope in how AI can and will transform medicine. And I hope to give you some ideas of what is possible, what is not possible, and where we are at today. Very briefly, a rather unusual slide for a scientist. This is in fact from Accenture analysis, a slide that shows what are the top 10 AI applications in health. And number one, with a value of $40 billion, is robot-assisted surgery, followed by virtual nursing assistants, administrative workflow assistants, fraud detection, dosage error reduction, connected machines, then identification of clinical trial participants, preliminary diagnosis, then comes automated image analysis, and then finally cybersecurity with a total volume of about $150 billion. What is the potential, or what are the main hopes where AI may bring us forward? This is, on the one hand, AI as a support tool for medical doctors to support clinical decision making, to complement human expertise, for example, in radiology, to provide access to current information. And you can imagine that a computer is very good at browsing information and providing information from scientific literature, from medical practice guidelines, etc. And that makes it particularly helpful and supportive for younger, less experienced doctors. And obviously, such a system has 24/7 availability. AI can help in early diagnosis, can predict disease outbreak, disease progression, and also treatment outcome, can provide feedback for patients and doctors, for example, concerning patients' compliance, and may help to reduce diagnostic and therapeutic error, thus increasing patient safety and at the same time saving costs. And last but not least, AI can extract information from huge patient numbers. And if you think about it, any doctor can only see a certain number of patients during his lifetime, but an AI can access data from all patients worldwide, basically. Here are a couple of successful applications of pattern recognition now in medicine. So, for example, the detection of diabetic retinopathy has been approved by the FDA for AI-based retina scan diagnostics. And at the same time, the same method was used also to classify gender. This was a side effect, just an incidental finding that based on retina scans, you can predict with 97% accuracy whether the person whom the scan is from is male or female. And just for comparison, a human doctor comes to a classification accuracy of 50%, so this is as good as guessing. Yeah, processing of images from radiology is one area where AI is very successfully used. And this is both MRI and CT scans. There are also a number of examples in bioinformatics, for example, gene signatures for breast cancer prognosis. But there are also some critical examples. So, for example, to predict suicidality or depression from social media profiles, an application that Facebook developed and then retracted a couple of years ago. Yeah, and I would like to take you through 3 more specific examples from our own work. The first example is an example that's more on the epidemiological basic research side, looking at molecular mechanisms of aging. The second example is one where we're looking for risk factors for fatty liver disease. And the third example is an example that's really in the clinic, predicting outcome for patients with cancer. The aging example that I would like to start with is very simply explained with this slide here. So the idea is to start with an old person, and then we want to apply some compound to this old person, and then somehow rejuvenate this person. And there's a reason why I'm putting Nivea here, and that is this is a joint project with Bayersdorf, the company that's producing Nivea, and also with the German Cancer Research Center. Yeah, very briefly, what have we done? We took skin samples from the epidermis, from the outer forearm of 108 healthy female Caucasian donors of different ages, 24 young individuals, 24 old individuals. Old here refers to 61 to 78 years. Young is 18 to 27 years. Children have been excluded because we did not want to focus on developmental genes. And then we had 60 participants in the intermediate range, so between 27 and 61. And for these participants, we took suction blisters, so samples from the epidermis, and then used these samples, put these samples through deep sequencing, both RNA as well as methylation, Illumina Infinium methylation microarrays, gene expression microarrays, yeah, and then also phenotypic data measuring things like skin elasticity, wrinkle depth, and similar properties. And then we trained, here in this case, a support vector machine, a fairly well-known machine learning tool, on the methylation data, on the microarray data. And this is the Illumina chip containing 450,000 CpGs. And the machine was trained to predict, based on these methylation profiles, to predict the age of the person. So in essence, you take the support vector machine, you tell it how old a person is chronologically, you give it the methylation profile, and then it learns to correlate the methylation data to the chronological age. And this was done with a linear support vector machine in a cross-validation way. So part of the samples is used for training, and a different subset of the study participants is used to make the predictions. And then this is run several times with different subsets of the data used for training and for predictions. And this way you can make an unbiased prediction for every participant. Yeah? And then the predicted age is interpreted because it's based on methylation data. The interpretation is that this reflects in some way the biological age of the participant. And then we are in particular interested in looking at outliers. So people that look biologically older or biologically younger than they actually are. Yeah? And at the same time, we can then look at molecular hallmarks of aging. Okay, yeah, and we did this on methylation profiles and also on gene expression data. And the first take-home message is that prediction of chronological age works much better based on methylation profiles than it does on gene expression data. For methylation data, we get an R-squared of about 0.95. For the gene expression data of approximately 0.8, with an absolute error for the methylation data of 6.5 years and an absolute error in the prediction for the gene expression data of 9.5 years. This is not overly surprising because gene expression data reacts more to environmental influences, whereas methylation in a way more reflects the actual underlying more consistent changes and not the immediate response to environmental challenges. Okay, yeah, and then using this methylation biological age predictor, Beiersdorf was particularly interested in individuals that have prematurely aged or that have managed to stay young. And the question, of course, is if someone like this person here is predicted to be, based on his biological profile, to be somewhere between 45 and 50 years old, but in reality this person is, well, almost 70, then the question is, what makes this person be younger biologically than he is chronologically? And yeah, we then also checked if this difference between the predicted and chronological age correlates to phenotypic properties of the study participants. And here, shown on the y-axis, is the difference between the predicted and the chronological age. So -10 here means the person is predicted, let's say 40 years, but actually is 50 years old. Whereas someone up here, +15, means this person is biologically 15 years older than he is chronologically. And if we correlate this with, for example, a questionnaire where people have been asked whether they have a lot of sun exposure, then you can nicely see that people with a lot of sun exposure are, in comparison to those with little or no sun exposure, prematurely aged. So this makes sense. Yeah, and then we looked at omics signatures. So the question now is, what are the factors that make the support vector machine predict that a particular person is very old in comparison to his true age, or much younger biologically in comparison to his chronological age. And then we essentially map the different genes to pathways. And this is shown here on this slide. You can see that there are particular processes that are coming up that we are now looking at together with Beiersdorf in more detail. Okay. The second project that I would like to talk about now takes you to the clinic. We're looking at lung cancers in smokers and how to treat these patients. And I would like to illustrate the question with a particular example. This is a 72-year-old male patient, a heavy smoker, 50 pack years. And he was diagnosed in 2014, shown up here, with a squamous cell carcinoma in the oral cavity. And this was treated, but 2 years later, the patient came back with a solitary nodule in the lung, again with a squamous cell carcinoma histology. And now for the treating physician, the question is whether this is a primary lung cancer. This would be good. Then you would basically operate this patient, and there's a good chance that you can actually cure him. Or is this lung tumor a metastasis of the previous squamous cell carcinoma? In this case, the patient would be stage 4, and you would essentially throw everything you have at him, but he would very likely still die. So the treatment option may actually be palliative chemotherapy. And for the pathologist, these two cases are extremely difficult to differentiate. And this is where we then came in with a mass spectrometer, taking the tumor sample, putting it through a mass spectrometer, and then analyzing the tumor this way, and again using a machine learning tool to predict, based on the proteomics profiles, whether the tumor we are looking at is a primary tumor or whether it's a metastasis. And what you see here is a decision tree that has been trained on this proteomics data. So basically you start with this protein here, and if the concentration of this protein is below a certain threshold, you go into the left branch. If it's above a certain threshold, you go into the right branch here. And then you look at the second protein and proceed similarly. And at some point, you end down here in a leaf of the tree. And these leaves contain probabilities telling you what's the probability for primary cancer versus for metastasis. And this is then very useful for the treating physician because he gets not only a diagnosis, but also an associated probability for the diagnosis. Yeah, and in fact, one of the curious findings here is that it doesn't really matter what machine learning tool you use. We tested a number of different methods here, and they all come up, with this exception maybe, with pretty much similar results. So the art is not so much in running the machine learning tool, but the difficulty really is in pre-processing of the data. That's where the quality of the ensuing classification is decided. We then compared our machine learning method with the classification done by a pathologist. And this is shown up here. The upper plot here shows, it's a Kaplan-Meier plot, for overall survival times of our patients. And the patients are grouped by the classification that the pathologist has done. Yeah? So the blue curve here is the survival curve for patients where the pathologist said this is a primary cancer, and the red curve is the survival curve for patients where the pathologist said this is a metastasis. And as you can see, they heavily overlap. So the pathologist is not really able to differentiate these two cases because we would expect that the primary tumors have a much better survival. And this is the outcome of the proteomic classification. So here you can nicely see blue is the group where the machine learning method said this is a primary tumor, and red is where the machine learning method said this is a metastasis. And again, this is done in the cross-validation way. You can nicely see again how the machine can nicely separate the two groups. Okay, and that brings me to the third example that I would like to present. This example now is one that comes from epidemiology, and we are now looking at risk factors for liver diseases. And in particular, we are looking at non-alcoholic fatty liver disease. This is a disease that starts with accumulation of fat in the liver and then progresses in 10 to 30% of the cases. Patients usually in the early stages show no signs and symptoms of the disease. Sometimes they are a bit tired, have some discomfort in the upper right abdomen, but nothing severe really. But in the long run, this leads to quite severe liver disease, ultimately cirrhosis and liver cancer. The disease is severely underdiagnosed. It's estimated that in the northeastern part of Germany, in the region where I'm working, the prevalence is about 25%. So 1 in 4 persons has fatty liver disease at different stages. Yeah. And the major challenge here is that there really is no non-invasive diagnostic method that's suitable for large-scale screening. And the diagnosis, really, the gold standard is still liver biopsy. But this is not something you can routinely offer for population screening. Yeah, and this brings me to the SHIP study. This is a large epidemiological study that's carried out in Greifswald. And as I said already, fatty liver disease has a high prevalence in the region, and this is the reason why. It's food, alcohol, and the result is that people are obese here and correspondingly develop metabolic diseases. And SHIP, the study of health in Pomerania, is a population-based study that looks for risk factors in northern Pomerania. And this is the region where sampling is done, up here, Greifswald and Stralsund. And the study started in 1997 and recruited 4,308 participants of age between 20 and 79 years. And then these participants have been re-invited every 5 years and are undergoing a quite extensive examination program. And this is shown here. So the study started in 1997 with 4,300 participants, 5-year follow-up between 2002 and 2006. Included 3,300 of these original 4,308 participants. Then the 10-year follow-up between 2008 and 2012, and the 15-year follow-up between 2014 and 2018. And still including 1,718 of the original 4,308 study participants. And in parallel, a second cohort was started in 2008 to 2012, SHIP Trend 0, in the same population, but it's a completely independent group of study participants, so there's no overlap. And a second set of 4,420 participants was invited. And the Trend 1, the 7-year follow-up examination, has been completed now. And this data is available now as well. So we have a second cohort that can serve as a validation cohort that has been examined with the very same methods and procedures. Yeah, and the examination program is quite extensive. So in the SHIP Trend 1 examination, participants spent over 24 hours in the examination center, including a sleep laboratory, for example. Whole body MRI measurements are done, ultrasounds, and they basically go through the different clinical departments and are examined from head to toe. And there's also a lot of molecular data gathered in SHIP Trend. This includes molecular data. We have genetic data from all participants. We have gene expression profiles in blood, proteomics data in blood, metabolomics data in plasma and urine, microbiome data from stool. And there's also still a lot of biomaterials stored, whole blood and plasma, nasal swabs, urine, stool, saliva, and also suction blisters from skin. And this is the data trove, basically, that my group now is working with. And we are then looking at integrating these molecular and clinical data sets and link them to development of disease over time. And we can go back 15 years to the initial examination, 20 years back, look at study participants 20 years ago, and then correlate what they looked like 20 years ago with diseases that developed later on. And this is ideal for machine learning tools to learn predictive models that can predict who will develop disease in the future and develop signatures that are suitable for early prediction also for other patients. So the idea here, and we used Bayesian networks in this example, the idea is to learn a Bayesian network on the chip data to model all interactions of processes inside and outside of the liver, which possibly drive non-alcoholic fatty liver disease. So basically, this proceeds as follows. We have the SHIP dataset, and this has incomplete because there are always missing values, some questions that weren't answered by a participant, or some molecular measurements that didn't work out. So we have to deal with this incompleteness of data. Then we discretize the data. An imputation needs to be done to handle missing values, and then the Bayesian network is trained on the resulting data. And this means if we have n different measurements, then there are n times n minus 1 divided by 2 possible interactions between these variables. So quite a lot of interactions if you think how many variables there are in a study like SHIP, including all these molecular data sets. And the distribution of the data makes things further difficult because we have a large number of variables, a relatively small number of study participants. So in mathematics, this is called the P is larger than n problem. We have a mix of discrete and continuously distributed variables, so measurements like blood pressure or height or weight that are measured on a continuous scale. Other questions that have yes/no answers. The continuous variables are of different distributions, not all normally distributed. There are things that can take on discrete values. A trivial example would be hair color. That can be black, brown, red, blonde. Variables are at different scales. Some variables are contain small numbers, others large numbers. Some variables are in micromole per liter, others are in kilograms. We have a large number of missing values, highly correlated variables, and also high levels of noise in the data. And that's a typical setting for epidemiological data, epidemiological study data, and it gets worse if you move to the clinic and look at routine clinical data. And that makes applying these machine learning methods complicated, but also interesting for us at the same time. Okay, and what do we want to do? We want to learn a Bayesian network from this data. What is a Bayesian network? If you look at this example here, we have, this is a small example, 3 different variables. B influences A and C, A influences C. And here in this case, we assume each of the variables can just be on or off. And there are certain probabilities. So B has a probability of 20% to be on and a probability of 80% to be off. B influences A, and if B is on, then A has a 20% probability of being on and an 80% probability of being off. Or if B is off, then A has a 1% probability of being on and a 99% probability of being off. And the same for C, except that C now depends on A and B. And the problem that we are now looking at is we do not know this network, we do not know the connections, we only know the involved variables, but we have observational data. And the question now is, from a table like this, where A may be fatty liver disease, B may be eating behavior, C may be some molecular marker, can we learn how these variables depend upon one another on the one hand, so the topology here, where are the arrows, and can we learn these probability distributions, just having this study data available. So two independent questions that need to be solved: find the model topology and find the model parameters. And for the mathematically inclined, this is done using Bayes' theorem, sampling either from a likelihood or from a posterior distribution. We in fact use the posterior here, and this allows it to include prior knowledge. And the trick here is that we have a very large number of variables. We have a relatively small number in comparison of study participants. So we need to penalize model complexity. And that's shown on this slide here. Basically, we favor through this prior distribution, and I don't want to go into the mathematical details here, but basically we favor models that have few connections. Yeah, so we do not assume that fatty liver disease is influenced by 10,000 other variables, but that it's a handful of variables that has an influence on fatty liver disease or on any other variable in the network. Yeah, and in order to do with the high dimensionality of the data, and by high dimensionality, again, I mean the large number of different measurements that have been done per study participant, and in comparison, the relatively small number of participants, we use a method here that we have adapted from hydrodynamics, and that's adaptive grid refinement. What's the idea in adaptive grid refinement? The idea is to improve the accuracy of a solution in a certain region that's of interest by adapting the granularity of the simulation in this region. So you start with a very coarse model. You look at what are the interesting regions, and in these interesting regions, you refine your model. And this is an example from, or this is a method that comes from hydrodynamics, from fluid dynamics. So we are talking partial differential equations and solving these PDEs. And except, yeah, as I said here, you start computing, and this is here an airplane wing, and you can see the wing here and how the flow goes, airflow goes over this wing, and you start this computation on a relatively coarse global grid, and then you apply an error estimator at every point in the grid, and then you look at your flag points where a lot seems to be happening and create a finer grid around these points, and in regions where nothing is happening or not much is happening, you do not refine. And then you refine the computation only in areas of interest. And this is iteratively performed until all error estimates fall below a fixed threshold. And we do a very similar thing to the Bayesian networks. We start learning a network between a small number of coarse clusters. So we cluster the different variables together and learn interactions between the clusters. And then we try to estimate an error in the clusters, flag clusters with high errors, and split clusters that have an high error up, and refine them. Yeah. Adding new clusters to the data, more refined clusters, deleting the larger ones, and then refining the network around the new clusters. So it starts at the bottom here with all the different variables, but we do not know the interactions between them yet. We cluster them together, then we learn interactions between the clusters, and then you refine the clusters in those regions where something appears to be happening that's related to the disease we are looking at, and then get a refined network and go back further until the model is completed. And this refinement, as I said, is done taking relevant output variables into account. So we do not refine the network everywhere, but around the region liver disease that is of interest for our research question at hand. Yeah, and this is what we did with the SHIP data on fatty liver disease. And I'm showing here a small excerpt from the network that was learned on this data. And the outcome variable is here, MRT, STEA. This is diagnosis of steatosis done with the whole body MRT that has been done on the participants. Lacking a liver biopsy, because these are healthy people, obviously, mostly. We clearly couldn't do a liver biopsy, so this is the second best substitute for diagnosing non-alcoholic fatty liver disease that we had. And what you can see here is, so the network shown here is what the machine learning tool directly learned from the experimental data. And this orange area here is a cluster that we have labeled fat distribution, and it contains variables like BMI, waist to height ratio, body fat in percentages, and similar variables. And you can see that these are linked to having fatty liver disease. Glucose here is also another variable that's related to fat distribution, or was found to be related to fat distribution, and again linked to fatty liver disease. Downstream of fatty liver disease, we had several variables clustered together that relate to liver function tests, so ALAT, ASAT, GGT values, and these in turn were linked to cell damage variables. Also interesting down here is cholesterol. And the machine also discovered automatically that there's a relation between glucose uptake and diabetes. That diabetes is related to insulin. Yeah, and it also made the link from diabetes to heart tissue, heart problems. So this is, we think, quite interesting because there's zero clinical knowledge going into the machine learning tool, but this is all automatically learned from the data. And we can now apply this method to different diseases and see if the machine can come up with new predictions, new predictive models. And the nice aspect here is that you get directly back an interpretable model. And what we are working on right now is to refine the method further and to include also molecular data in these predictions. Yeah, and with that, I'm coming almost to the end of my presentation. I would just give you an outlook on other types of work that we do in the group. So, for example, the intraoperative classification of tissue type during brain cancer operation. This is based on infrared spectroscopy. So during the operation, an image is taken, and then based on the infrared spectroscopy, we train a machine learning method to classify the type of brain tissue and label where's tumor, where's healthy tissue. Another example is prediction of mortality using deep learning on chip data. Prediction of mortality based on electronic health record data. We are applying neural networks to predict cardiovascular disease based on ECG data. Then we've done a lot of work on predicting survival in cancer patients using penalized survival models. We are looking at, with these Bayesian networks, also risk factors for thyroid disease. We have worked on prediction of therapy response for pre-diabetic patients, and we are also starting to employ machine learning to predict depression based on social media data. And with that, I would like to conclude my presentation and thank you very much for your attention.